Did you know that your body kills cancer each day? It's true. Every day, cells in your body make mistakes, and those mistakes can lead to cancer if they're not taken care of. But if we kill cancer each day, then how do we get cancer? That's a question that researchers have been trying to answer since cancers were first discovered. And now we have a new tool providing us insights into the 3D structure of cells that provides additional information to help us treat cancer. Structures and the pieces that make up cells are highly important for how a cell works. Think of it like a building. You have walls, support beams, windows, doors, etc. But those pieces have to be arranged in a very specific manner to make a functioning building. Usually, we'll follow a blueprint when we're building. Cells do the same, but their blueprints are written in code as DNA. For today, you can just think of DNA as beads on a string. Traditionally, we'll look at the sequence of DNA, which is just essentially asking what order would the beads appear. Are they blue, blue, red? Red, blue, green? Green, green, green? Changing the sequence changes the way that the cell functions. However, the sequence only provides us so much information. It would be kind of like having a blueprint that only lists the materials you need rather than how to actually build it. For cancer, we can find out if the sequence has changed, but when we look at it, we find out that it actually hasn't for the most part. So where do we look next? And that's where the 3D structure of DNA comes in. In terms of our building, we're basically asking what kind of room does it make? Is it a bedroom or is it a kitchen? Each of those has 4 walls and a door or 2. Your cells have a similar structure. All of the cells in your body have the same sequence of the same parts, but your heart cells perform a different function than your kidney cells, just like you don't cook in your bedroom. If you were to cook in your bedroom, that would be a multitude of issues. You'd have rotting food, smoke detectors going off. It would be unpleasant at best. And that's basically how cancers work. Cancer cells perform the wrong function for the space that they're in, and that change in function can partially be attributed to which pieces are interacting and when. Let's think of these pieces more like locks and keys now. A key can go into a lock and allows it to open. In terms of DNA, a key is called an enhancer and a lock is called a promoter. The enhancer unlocks the promoter so it can express a particular trait. Enhancers and promoters have a lot of abilities that are really similar to locks and keys. There are certain keys that can unlock more than one lock, such as a key that unlocks both your front door and your back door. There are also locks that have more than one key, like your car has multiple keys you can use to drive it. Our job as scientists is to figure out which promoters are interacting with which enhancers at different times in different cell types. Cells don't do the same things all the time. In your kitchen, you'll cook at certain times and you'll clean at others. Cells can also switch tasks by changing their enhancer-promoter interactions. Cancers are a little tricky, though. When we look at their 3D interactions, we find that they've essentially figured out how to lockpick on a cellular scale. It's like cleaning in your kitchen when you should be cooking. They'll open things they shouldn't be able to, and that causes a lot of havoc. But if understanding cancer and cell operations is as simple as fitting locks into keys, then why aren't we done yet? It's not actually so simple. Imagine you have a pile of locks now, and you have a pile of keys. Nothing is labeled, and you have a toddler that keeps coming around and shaking up all of your piles. That's really closer to the question that we face. This new data type gives us a lot of new information about the structure, but we still have significant amounts of work to do to understand what we can measure and when the things that we measure as differences are normal differences or cancerous differences. But once we understand the fundamentals and how normal differences are and how cancerous differences are, we can start to make better treatments for cancer and even other diseases like asthma and Alzheimer's. But studies like this cost a significant amount of money. The government's responsible for allocating funds for scientific research, and you can help us by voting for politicians who support the funding of scientific research. It won't cost you anything, and the benefits will reach the entire world. We can't do it without you. Thank you.